In this video, I'm going to give you a basic overview of synapses and neurotransmitters. Before we get into the details, I want to talk about the kind of cognitive tasks that the brain needs to be able to do. Suppose, for example, that I showed you this sentence and I asked you to read it out loud. Jim plays baseball. Well, what would it take for your brain to do this? Well, we'd have to activate neurons in our eye in order to send that visual information. First, it would go to the thalamus, and from there it would project to a part of the cortex, the back part of the cortex called the occipital lobe. There are a number of outputs from the visual part of the cortex, but one of them goes to the temporal lobe, where there's a part of the brain that's involved in processing and understanding language. That sends projections to the frontal lobe, where there's another part of the brain that's involved in producing speech. We'd have to activate some of our motor cortex, the part of the brain that's involved in putting together a plan for us to activate muscles in a particular order. Those would project down to the brainstem, where there are the cell bodies of neurons that can go out and innervate the facial muscles. Speaking also involves controlling our breathing, and so we'd have to activate muscles in our chest as well, so there'd be even more. There are a couple things I want you to take away from this. The first is that different parts of the brain are specialized to carry out different functions. I said that the back of the cortex was involved in processing visual information, that there was a language comprehension center, that there was a speech production center, that we had a part of the brain that was involved in the initiation of movement, and that there would be cells that could send out motor instructions to muscles in the brainstem. Well, if you think about it, this modular arrangement makes sense. We rely on our visual system for many things, not just reading words, so we might want to have other processes that get sent to the occipital lobe for visual processing. We might want to use language comprehension not just for reading words, but also when someone speaks to us so we can understand and partake in a conversation. And we might want to initiate movement and activate muscles in our face if we were going to eat or chew or smile. So having this modular arrangement of the brain that we can link up by circuits makes a lot of sense. Well, let me pull these away just so I can talk about the other thing that I want to point out. And that's that because we want to have a modular arrangement, we rely on having nerve cells travel from one brain area where processing occurs, and then that information gets sent to the next, to the next, and to the next. And at each one of these spots here, there's going to have to be communication, where a signaling cell is going to have to signal a recipient cell, and that's going to occur at these structures called synapses. So there are going to be nerve cells that are going to be releasing a neurotransmitter onto a target cell in order to send information through this pathway. We'll spend the rest of the video talking about the details of how synaptic signaling happens. And what I want to tell you is we're really only going to be looking at a tiny fragment of the signaling cell and the recipient cell, just at the part where they're communicating with one another. Let's begin with some labels. I'm going to start by telling you about what's called an excitatory synapse. That's when the signaling cell signals the receiving cell and gets it to fire a nerve impulse too. In my cartoon, the signaling cell is on the left, it's gray. Receiving cell will be in yellow on the right. Almost all nerve signaling involves the release of chemicals from the signaling cell onto the receiving cell. And nerve cells take those chemicals, those neurotransmitters, and they package them into packets called vesicles. For most common neurotransmitters, in our case these blue stars, the neurotransmitter is synthesized at the axon terminal itself. Usually there's a precursor molecule that can be converted with the help of specific enzymes in order to produce the final product. And then once it's made, it doesn't stay loose in the cytoplasm. It gets packed into those vesicles so it'll be ready for release. Release occurs when a nerve impulse arrives at the terminal. And what happens then is the vesicles will fuse, releasing their contents, and the receiving cells have specific receptors for these molecules. When the neurotransmitter binds to its receptor molecule, these channels can open. Often signaling involves the movement of ions through these channels. In this case, for an excitatory synapse, what happens is the cell gets more positive, and that makes it more likely that the receiving cell will fire a nerve impulse. Excess neurotransmitter can be removed from the cleft using the help of special transporter proteins. In this case, we see it as a red protein. It'll pick up that, move it back into the presynaptic cell where it can be recycled. The last thing I'm going to tell you about this is that Although there are lots of different neurotransmitters, in the mammalian brain, the neurotransmitter at most excitatory synapses is a particular chemical. It's called glutamate. This is one of the two most common signaling molecules in the brain. In the previous example, I focused on a single signaling cell and a single receiving cell, but the situation in the brain is typically way more complicated. A single neuron often receives many presynaptic inputs. The neurons in your cortex receive about 30,000 synapses each. For many neurons, we can see the number of synaptic inputs that they have because they are spiny neurons. We see an example of this in this cartoon. What I want you to notice is that the dendrites of this are covered with short little spikes. Those are spines. And studies have shown that one spine is equal to one synaptic input. Now, the artist who drew this cartoon was not exaggerating the number of spines. In fact, if anything, they were underestimating it. Here's an image from an actual nerve cell. This was stained using specific probes that will bind either to the signaling cell or the receiving cell. In this case, we can see the cell body of the neuron. The blue indicates where the nucleus would be. And what I want you to see is that on the dendrites that are extending away from that cell body, there are many, many, many red speckles. Those red speckles are stained with a specific protein that's only found in axon terminals. So each one of those red dots indicates a different presynaptic input. You can imagine the amount of input that's coming into this cell. Adding to the complexity is that not all of the input coming into the cell is going to be excitatory. Sometimes the signaling cell will release a neurotransmitter and inhibit activity in the receiving cell. It will make it less likely to fire a nerve impulse. Another complication is that some inputs are short-lived, while others can have a long-term impact. The excitatory neurotransmitter that I told you about, glutamate, has immediate, short-lived effects, but other inputs to cells can be modulatory. They can exert their effects over minutes or hours. Let's take a quick look at some of these variations just so you see how they might work. Here's a cartoon of an inhibitory synapse. You can see that there's a signaling cell and a receiving cell, and in most ways everything looks the same. There might be a precursor molecule that needs to be converted into the neurotransmitter. It will have to be packaged into those vesicles. If a nerve impulse arrives, it will be released from the cell and it can bind to a receptor molecule. But here what will happen is that when the molecule binds to the neurotransmitter receptors, typically negative charges come in. And the cell gets more negative, and that's going to make it less likely to fire an action potential. In the mammalian brain, the transmitter that we find at most inhibitory synapses is Gaba. So a typical neuron in your brain has some receptors for glutamate, some receptors for Gaba, and whether or not that receiving cell is firing nerve impulses will depend on adding up all of the excitatory inputs and all of the inhibitory inputs and seeing if the cell can reach a threshold for itself being activated. If it doesn't reach that threshold, no nerve impulse will occur. The last thing I want to talk about are modulatory synapses, and here you can see I've added a third neuron. The bottom two neurons, the gray one and the yellow one, that's just the excitatory synapse I showed you before. You can imagine a glutamatergic cell releasing glutamate onto the receptor cell, and if that molecule binds, we might or might not have the yellow cell reach threshold and fire a nerve impulse. What modulatory synapses do is they can also form synapses onto those receiving cells. In this case, we've got a different neurotransmitter indicated by these pink triangles. If those pink triangles are released and they bind to the receptor molecule, we can activate cascades. Often this is something that's a little bit slow. There might be a number of steps involved. It might take seconds or minutes for this to kick in. One of the common things that can happen is that this series of events can go and it can modify receptor proteins, making them less likely or more likely to open. And what that means then is that the same input from the gray cell could either be strengthened or weakened depending on what kind of compound was released from the blue cell. It's modulating the strength of this synapse. These types of modulatory synapses are really important, and some of the most well-known neurotransmitters, dopamine, serotonin, histamine, norepinephrine, act primarily by modulating synapses where GABA or glutamate are the neurotransmitters. Well, I know I've covered a fair amount of information here, but the reason that I've done this is because most drugs that affect the nervous system, and I'm talking about street drugs here, but also pharmaceuticals, exert their actions by targeting one of the proteins that I've colored here. The blue receptor, or the red or yellow reuptake transporters, or the green receptor, those are all potential targets for pharmaceuticals that can modify brain function. As we continue to look at the brain and at conditions that affect nerve functioning, we'll come back and we'll see many of these players again.